If your actions create a legacy that inspires others to dream more, to learn more, and to become more, then you are an excellent leader. Good morning, everyone. I, Dr. Sakshi Bhardwaj, on behalf of Altum Technologies, I welcome you all in our Women's Day special webinar series, specially dedicated to all the women researchers out there. We all know a fact that vitamin D, a human body only produce vitamin D when the one is exposed to the sunlight, right? Vitamin D has several important functions. Perhaps the most vital are regulating the absorption of calcium and phosphorus and facilitating the normal immune system functions. But unfortunately, we all are not getting that exact amount of vitamin D, right? So, which we are supposed to be, okay? And hence, it may cause many disease conditions, right? Vitamin D deficiency is a global concern actually. So, the topic for our today's webinar series is in silico study of polyunsaturated fatty acids as potential modulator of wild and mutant vitamin D receptors. Our guest of the honor for today's talk will be explaining about how selective polyunsaturated fatty acids would modulate the dynamic and kinetics of vitamin D receptors irrespective the bioactive of vitamin D binding. It's giving me immense pleasure to introduce our speaker, Dr. S. Jubie Jawahar, Associate Professor, Department of Pharmaceutical Chemistry, JSS College of Pharmacy, UT. She has done her PhD from JSS Academy of Higher Education and Research, Mysore. She is having 21 years of teaching and research experience. Her research area of interest are discovery of novel chemical entities for the cancer therapy through the various in silico tools and drug repurposing. Okay. She has been involved actively in the isolation of fatty acids and synthesis of novel bioactive molecules for the cancer therapy. She has been the committee member of NAC, NBA, NIRF, and many other professional bodies. She has published 61 international and 29 national articles with all the good impact factors. Her total citation are 1,461. She has 4 Indian patents too. She has published 5 books and 2 book chapters. She has been sponsored by many government bodies for their grants. She has guided many PhD and MPharm students and various pharma projects too. She is a recipient of Young Researcher Award 2019 and Young Performer of the Year 2022 from the Association of Pharmacy Professional India and got the Best Researcher Award 2019 from the JSS Mysuru. Nevertheless, her biography has been included in famous India nations Who's is Who. So, and now today, without any further ado, let's start. It's up to you, ma'am. Welcome you once again.
Yeah, thank you, Dr. Sakshi, for your nice introduction. Indeed, it's a pleasure for me to be a part of this Women's Day program, and it gives me an immense pleasure to be a woman and to be an active researcher in the field of bioinformatics and pharmaceutical research. And in next 45 minutes I'll be talking about the in silico study of polyunsaturated fatty acids as potential modulators of wild and mutant vitamin D receptor. So my presentation goes like this. The first will be genesis of the work, then how we formulated our hypothesis, and followed by that what are all our objectives and how we have examined our objectives by using the in silico tools by BioVR Discovery Studio, molecular docking, in silico ADME predictions, molecular dynamics, and it will be ending with the conclusion. Yeah. So, as Dr. Sakshi told, the vitamin D, it is a sunshine molecule. It is indeed getting lots of attention due to the alarming rise in the prevalence of vitamin D deficiency. Especially this vitamin D, it is closely associating with the metabolic disorder type 2 diabetes mellitus. And so many clinical studies and prospective studies demonstrate that there is the inverse associations of vitamin D plasma concentration with the risk of type 2 diabetes mellitus. Not only that, this vitamin D deficiency is also reaching epidemic proportions in India, with the prevalence exceeding 70% of the total population. Yeah. So the molecule of interest, that is particularly vitamin D, it is nothing but 1 alpha 25 dihydroxy. That is 1 alpha 25 dihydroxy vitamin D3. It is a fat soluble steroid hormone which is considered as a pivotal role in various biological process, including skeletal muscle development, insulin sensitivity, glucose utilization, and wasting. And there is a interesting study that the supplementation of vitamin D in insulin resistant or pre-diabetic individuals, they have shown improved insulin sensitivity. And one of the recent reviews, okay, they have conducted in 137 healthy volunteers. Okay, so there also they have observed there is a strong association of this vitamin D level in plasma with metabolic and other chronic diseases. So based on this, this vitamin D as a predictor and biomarker for micro and macrovascular complications in type 2 diabetic individuals. Not only in type 2 diabetics mellitus, this vitamin D is also playing a critical role in skeletal muscle development, glucose homeostasis, and wasting. Okay, and so all these evidence indicate there is a strong association between vitamin D levels and metabolic health and skeletal muscle functions. Okay, so what we observed that the absence of any remarkable reversal with vitamin D supplementation strongly warrants an extensive in-depth understanding of molecular regulation and dynamics of vitamin D signaling. That is, how the vitamin D is positioning in the vitamin D receptor, and the three-dimensional arrangement and three-dimensional conformations are directly related to these diseases. That's what we have observed our extensive literature review. So coming to the vitamin D receptor, so it is a nuclear receptor and it is found in prostate, ovary, breast, skin, brain, heart, pancreas, kidney, intestine, and colon. So it shares the main structural characteristics. It consists of a highly conserved DNA binding domain. It consists of a highly conserved DNA binding domain and ligand binding domain, a hinge region which connects the DNA binding domain and ligand binding domain, and we have an activation factor 2 also. As I told you, it contains two domains, and the DBD, that is the DNA binding domain, it contains two zinc fingers, which is having 4 cysteine residues in the tetrahedral arrangement. And the ligand binding domain of this VDR, it has got a ligand dependent activation function AF2, which is a major interface for homo and hetero dimerization and interface for various co-activators as well as co-repressors. So this is the structure as I told you, and this is the occupation of the vitamin D, which is positioned in the vitamin D receptor. So, the dynamics of vitamin D receptor activation in the presence of various orthosteric or allosteric ligands in both skeletal and non skeletal systems are gaining tremendous scientific attention momentarily. And coming to the signaling of this VDR. So what happens, this vitamin D, the moment it binds to the VDR, it goes and binds with a receptor which is called as 9 cis retinoic acid receptor, that is RXR. So this RXR and VDR forms the heterodimerization complex, and they initiate the VDREs, that is vitamin D response elements, which recruit the target genes. And this interaction intrinsically leads to the regulation of calcium, phosphate, and other second messenger pathways. So this is the complex of X-ray crystallographic structure of VDR and RXR complex, and this is the cryo electron microscopy complex of the VDR and RXR. So this VDR and RXR complex is very important for the recruitment of various target genes via the recognition of the VDR response elements. So now coming to the mutations. So as I told you, there is two domains, DNA binding domain and ligand binding domain. So the mutations in the DNA binding domain of the VDR interfere with VDR RNA interactions, but significantly it won't do any ligand binding and result in loss of VDR function. But interestingly, if any mutation occurs in ligand binding domain of the VDR, it do affect the heterodimerization with RXR retinoid receptor, and it inhibit the coactivator interactions. So as the outcome, these ligand binding domain mutations, it results in partial or total hormone unresponsiveness. So the mutations in the ligand binding domain may be the major cause for the hormones unresponsiveness. Okay, and few published reports demonstrate the association of metabolic diseases with a few single nucleotide polymorphisms. So among the single nucleotide polymorphisms, these are the SNPs, that is BSM1, TAC1, APA1, and FOC1. So these SNPs were associated with the type 2 diabetes mellitus, insulin resistance, and muscle strength. Now coming to the polyunsaturated fatty acids. Okay, so where we have got the lead how this polyunsaturated fatty acids modulates the vitamin D receptor. So the researchers Jurupta and colleagues, they only first demonstrated the omega 3 and omega 6 polyunsaturated fatty acids like EPA, eicosapentaenoic acid, and DHA, docosahexaenoic acid, and omega 6 fatty acid, arachidonic acid. So they bind with low affinity to the ligand binding domain of the VDR. So what they do, they lead to this VDR-RXR complex formation and the transcriptional activation. So it is likely that this vitamin D and polyunsaturated fatty acids, they have the complementary roles. And so many experimental and clinical studies, they strongly support the pivotal roles of this PUFAs in insulin sensitivity and glucose utilization. And this is an interesting study, meta-analysis conducted on middle-aged overweight men shows that the participant with the higher omega-3 intake, the participant with higher omega-3 fatty acids intake, they have higher insulin sensitivity and improved metabolic profile. Not only that, the consumption of the short-term fish oil, which is extensively having the omega-3 and omega-6 fatty acids, so they also increases the insulin sensitivity in people with metabolic disorders. So these findings established the scope for these PUFAs as effective VDR agonists or modulators, and we could not find any in silico supporting paper for this PUFA or modulating this VDR. Yeah, so as I told you, so this will be the eicosapentaenoic acid. This is a omega-3 fatty acid, which is extensively present in cod liver or fish oils. And this will be the docosahexaenoic acid. Again, this is also omega-3 fatty acid. Okay, and this will be the omega-6 fatty acid. We know very well the arachidonic acid. It is directly connected to the prostaglandin, eicosanoids, and inflammatory markers and inflammatory pathway. So these three fatty acids, PUFAs only, we have selected for our study how these three fatty acids are modulating the vitamin D receptor. And coming to the hypothesis of the study, so as you all see here, so this will be the vitamin D receptor, and the one, it indicates the binding site of vitamin D on the vitamin D receptor. So the moment the vitamin D binds to the vitamin D receptor, so it goes and binds with RXR, and it forms the complex VDR and RXR. Then this VDR and RXR complex bind to the gene packet and alter ligand mediated transcription. Simultaneously, this long chain fatty acids, okay, so they have the low affinity towards this vitamin D receptor, binding of this PUFAs like EPA, DHA, AA to initiate the reaction steps. The moment it goes and binds to this packet of this VDR, so as the vitamin D does, all the subsequent steps will be followed by this PUFAs. On the right side, in case any mutations occur in the vitamin D, or that is vitamin D receptor ligand binding site, mutations in VDR may definitely alter the structure of the receptor, leading to altered patterns of binding of long chain fatty acids and vitamin D. So, what is our proposed hypothesis? The long chain PUFAs, for instance, this EPA, DHA, and AA, would directly modulate the dynamics and kinetics of VDRs, irrespective of the bioactive vitamin D binding to VDR. That is our hypothesis. So, in order to establish the support for our hypothesis, we have formulated the following objectives, and we have experimented using in silico studies. So the first one is, of course, we have taken three types of VDRs. The first one is the wild type VDR, where the vitamin D is positioned. Study the binding affinity of the PUFAs on wild type VDR. The second one is the VDR, which is having a co-activator peptide. Examine whether these PUFAs influence the transcriptional changes on VDR with peptides as specific co-activator. Then, of course, we have taken a mutated VDR, and we wanted to check whether these PUFAs will be modulating the VDR also. So for the examination, we have used the C-Docker module and molecular dynamics module, ADMET modules of Discovery Studio to understand and elucidate the proposed hypothetical mechanism. So as I told you, these are the three VDRs we have selected. The first one is the wild type, where the VDR is deeply buried in receptor, which enables very specific interactions with natural ligands. Most prominently, the 1-alpha 25 dihydroxy vitamin D3. The PDB ID is 2ZLC. And the second one is the VDR, which is having the short co-activator peptide LX like that, and it is the PDB ID is 1 rkg. And the mutated receptor, actually right now these two mutations, that is with the crystal structures are available, R270L and W282 are mutants, where arginine is replaced with leucine and tryptophan is replaced with arginine, which correspond to the human R274L and W286 are mutants. So these are are the three proteins we have taken for our in silico study. So majorly we have performed the molecular docking study, and which is supported by the molecular dynamics study. And the molecular docking studies have been carried out by using Discovery Studio, that is C Docker module. So we know very well. So here the protein will be like the ligands will be very flexible, and the protein will be rigid. And the minimized structures of the selected polyunsaturated fatty acid, the three acid DHA and EPA, AA were taken as the input ligands, and C Docker energies were computed. We have studied the amino acid interactions between the ligands and protein. And we know very well the charm force field was used to minimize the energy. And the docking protocol C Docker charm scheme to dock ligands into the receptor binding site was applied. Then random ligand conformations were generated using high temperature MD. Then the conformations obtained were translated into the binding site, and compound poses was created using random rigid body rotations followed by simulated annealing. Then the final energy minimization was used to refine the ligand poses, and we know very well the C Docker energy and C Docker interaction energy have been taken. And what is C Docker energy? It is sum of the internal ligand strain energy and the receptor ligand non-bonded interaction energy. And C Docker interaction energy is nothing but the non-bonded interactions between ligand and receptor were computed for each pose. The pose with the lowest energy values indicated the most favorable binding model. And these are the outcomes of the molecular docking. Yeah, before getting into the molecular docking protocol, all the VDRs, that is vitamin D receptors, were validated by Ramachandran plots, which revealed that most of the residues were in the favored regions. Okay, and of course I didn't show the ADME results also. All they are in the limit, all three fatty acids. So we can see the binding energy, C Docker energy, and C Docker interaction energy in the table. So we can see very well that compared to the vitamin D, the binding energy you can see here minus 78.79 against the wild type VDR. So our compounds are having a significant binding affinity, okay, in terms of the binding energies, okay. And not only to the 2ZLC, that is wild type, and to the with the coactivated also mutant VDR also, all our fatty acids are having significant binding energies when compared to the vitamin D itself. So by this binding energy calculation, we came up with the preliminary conclusion that the selected PUFAs appreciably modulate the vitamin D receptors. To support this one, we have extensively studied the two-dimensional interactions. Okay, so the increased binding affinities of PUFAs were further supported by the comparative analysis of receptor ligand interactions with the core amino acid residues. So as we all know very clear, usually we observe the non-covalent interactions such as hydrogen bond, hydrophobic and electrostatic between the PUFAs of the ligand binding domain and VDRs. So here, the native ligand vitamin D3 exhibited hydrophobic interactions such as alkyl and pi alkyl with the catalytic residues of the VDRs, whereas all the PUFAs has additional van der Waals interactions and electrostatic interactions too. As we all know very well, these polyunsaturated fatty acids, they have a long alkyl or alkenyl chain. So of course, they do undergo these van der Waals and electrostatic interactions. And this carboxylic acid group is also a major one of the reason it undergoes major hydrogen bond interactions compared to the vitamin D itself. So the electrostatic and van der Waals interactions were observed more when compared to the native vitamin D ligand. Similarly, not only to the wild type receptor, towards you can see here for the coactivated peptide also, and for the mutated receptor also. And what is our understanding, we also studied the binding modes of all the three PUFAs in the binding packet of the VDRs tested. So we know very well the any agonist or antagonist and its binding affinity, it is basically depending upon the nature of the core structure of the ligands, nature of the chemical signature, what type of moiety, what type of ring system is present. It certainly it affects the binding affinity. So here we can see very well, there is no connection between or no structural similarity between this vitamin D as well as the PUFAs, because the chemical signatures are completely different. Vitamin D, it has got a ring aromatic, and the PUFAs, it is having long alkyl and alkenyl chain. Okay, so it has the highest binding affinity when compared to the vitamin D3. Maybe the number of rotatable bonds present in the PUFAs, it may play a major role for this increased binding affinity. And we know very well that the presence of greatest number of rotatable bonds generates more conformations, which leads to more freedom in the binding site of the receptor. So that's what our observation from these molecular docking studies. And after that, the molecular docking studies, then we have moved to molecular dynamics. So here the highly constructive conformation poses and favorable bonded complexes form receptor ligand interaction, have used an input to study molecular dynamics simulation using BioVR Discovery Studio. Again, the charm force field was applied to two-step minimizations of 500 cycles with a steep descent and conjugate gradient, and the relaxed and energy minimized complex was then subjected to three steps of dynamics. And it was the receptor initially the receptor ligand complex was gradually simulated and heated from a temperature of 50 Kelvin, followed by its ramping to 300 Kelvin, and after that it is equilibrated by 100 steps. I think these are the standard protocol, and we have gone for 500 picoseconds, that is 5 nanoseconds. And we have observed the parameters RMSD, root mean square deviations, and radius of gyration RG, and hydrogen bond. We have observed what are all the hydrogen bond interactions after this dynamics, and every 2 picoseconds it was observed. And coming to the outcomes, so we have taken the mutated VDR, that is 3VTC with DHA complex. We have chosen, we have taken for our molecular dynamics studies. Okay, then RMSD values of each receptor ligand complex were plotted to elucidate the effect of the compounds on the structural stability and integrity. So we can observe here initially, maybe this is our compound, the DHA with 3VTC, and this is the vitamin D with 3VTC. We can see here, maybe in case of vitamin D, up to this 0.75, up to 0.75, that is up to 1700 or 1800 picoseconds, there is a slight what I can say, slight increase until 5000 picoseconds. It was in the stable rate, that is it is in the range. You can observe here both the complexes. That's what we observed. So from this plot, the 3VTC vitamin D and the compound showed a sharp increase in RMSD up to few picoseconds. Later, the deviations were optimized up to 1700, then increased slightly to 0.75 angstrom, and the complex attained equilibrium. That's what it is. It is attained equilibrium till the end of dynamics. So similarly, the DHA complexes also, it showed a fluctuation up to 0.89 up to 1000 picoseconds. Then it attained the equilibrium till the 5000 picoseconds. So the results. So overall, both complexes maintain the equilibrium with minor changes in the conformations. Then the second parameter is observed. It is radiation radius of gyration. So we know very well it estimates the compactness of protein folding and unfolding mechanism throughout the dynamics. So here the RG values of 3VTC with DHA initially started from 18.82. Then the structure showed a gradual increase of RG reaching 18.83 angstrom, and short plummet at around zero point 0.5 nanoseconds and increase gradually to attain equilibrium after 1 nanosecond. So you can see the up to 18.81 here, 18.81, then almost it is attaining the equilibrium. Similarly, like our vitamin D also. So the results indicate that 3-VTC DHA is aliphatic substituent to the protein binding site, showing significant changes with the higher RG value of 18.84 angstrom when compared with the reference vitamin D, which showed an average RG value of 18.79 angstrom during the 5 nanosecond of molecular dynamic simulations. Then afterward, the hydrogen bond profiling, it has been observed. So the residues, the crucial residues, constant binding interactions we have observed with the active amino acid residues, and all are in the range 1 to 2.5 angstrom. Similarly, the vitamin D also has like given the all the active site amino acid residues are involved in that interactions, and the range is 1 to 1.9 angstrom. You can see the pictures also. Yeah. So preliminarily, we have done the molecular docking, and it is supported by our molecular dynamics study also. So what is our observation, our findings are, the VDR coactivator binding site is formed when a VDR ligand is bound to the ligand binding domain of VDR. So for effective transcription, it is essential to suppress the different coactivators binding to the VDR coactivator binding site. So we have selected the VDR with the coactivator, so which is called as DRIP2, that is cap LXLL, where we know where L is leucine and X is any amino acid. The repositioning of helix H2 after vitamin D3 binding is critical for the formation of a hydrophobic groove that binds this DRIP2 motifs in the nuclear receptor interacting domains of coactivators. So it's our understanding that the successful interactions with hydrophobic residues, mainly with leucine, may enhance the effective binding. And why we have concern about this mutations or mutated receptor? Why? Because the hereditary vitamin D resistant rickets, that is HVDRR, so it is an autosomal recessive disease which is exclusively caused due to the mutations of the ligand binding domain of the VDR. Okay, so in particular, the missense mutations R274L and W286R in the ligand binding domain of the VDR can severely reduce or even eliminate natural hormone responsiveness. So mutations in the VDR LBD have been shown to interfere with some aspect of this vitamin D3 binding or VDR signaling. So the types of defects include ones that completely abolish, it may completely abolish the vitamin D binding to the receptor, or it reduce the affinity of vitamin D towards the VDR, or it disrupt the VDR-RXR heterodimerization complex formation, or it interfere with coactivator interactions. So any of these four possibilities. So 15 missense mutations have been identified in the ligand binding domain that reduce or abolish VDR functions such as ligand binding and ligand dependent transactivation. And so far, only the crystal structures of HVDRR H305Q or 274L W286R. These are the three mutants with the crystal structures so far have been identified. So among this three mutants, this R274L, it causes more than 1,000-fold reduction in responsiveness to the natural hormone. Similar way, this W286R also. So that's what we have chosen the VDR with these two mutations, R274L and W286R. So 10 patients with this mutation never respond to the natural hormone. So coming to the conclusion, so we have proposed the hypothesis that these PUFAs, especially EPA, DHA, and AA, they modulate the VDR. So the proposed hypothesis and supporting in silico evidence provide a concrete base of the process by which selective PUFAs influence VDR. This would be the definitive pioneering study of the complementary roles. So the outcomes will be applied to develop novel treatment or prevention strategy to combat vitamin D deficiency. But however, while we support our hypothesis with in silico approach, but definitely the outcome of the study should be supported by a real scenario with in vitro cell lines and animal models to support the proof of concept. So nonetheless, the elucidation of complementary roles of essential PUFAs in VDR activation establish the proof of concept for their potential modulatory role on this vitamin D receptor. And these whole findings are published in this mini reviews in medicinal chemistry. Yeah. So with this, I'll be acknowledging. I'll be concluding my talk, and my special thanks to my sincere pranams to His Holiness Swamiji of JSS Mutt and all the leadership of JSS Academy of Higher Education and Research, JSS College of Pharmacy and Mooti, and my special thanks to all my PG and PhD students. In fact, this work has been done by one of our student, Mr. Balaji, and you can see all my papers and publications in all these databases. And whatever I have gained, I have contributed a book, a textbook of computer aided drug design with one of my friend, Dr. Prabha, which is exclusively for the undergraduate and postgraduate pharmacy as well as other life sciences students. And my special thanks to this Altum Technologies Private Limited, and I have associated with them for I think more than 7 to 8 years. So it was a very good experience, and with their support, I think during COVID time they have given a free evaluation license, and we have done some work based on the green tea components, gallocatechin analogs, and we have published this work, and it has come in RSC Advances in Hot articles along with one of my collaborator, Dr. Suresh Kumar. Now he is in Swansea University, UK. So thank you very much for your patience. And once again, I thank the Altum Technologies for giving me an opportunity to share my study to the audience. Thank you, Dr. Sakshi.
Thank you so much, ma'am. It was very knowledgeable session. You have beautifully explained how the in silico predictions could be helpful not only for the synthetic components but of course for the natural ones also. How we can utilize these computational predictions to get a lead molecule, or how we can utilize these real time environment predictions to check the binding affinity for our whether it is a natural protein or a synthetic, whether it is a big one or a small one. You have told how the Discovery Bioware Studio you have utilized, and how this Bioware Discovery Studio is using a charm dynamics basically, which is very accurate to predict the results. So yes, we have some questions, ma'am, so it's time to take those questions.
Yeah, sure.
uh, The first one is why mutated VDR is compared with the wild VDR.
Yeah. So as I told you, basically all these metabolic disorders, especially type 2 diabetes mellitus and all, the mutations occurs in the ligand binding domain. Okay, due to the mutations, the hormone, the VDR, it becomes totally like it becomes what I can say. Unresponsiveness. Okay, so due to that we wanted to check whether these PUFAs are having effect on mutated VDRs also. So that's what it is, we have checked about the mutated VDRs.
Okay. Next question is, are there any changes in the binding sites was observed in between these three VDRs?
Binding sites, I think we didn't observe any major change in the binding site between these three VDRs.
Okay. Next one is any effects after saturation point of the receptors used?
uh, any effects after saturation point of the receptors used? Maybe I think Sakshi can answer this question. Saturation point of the receptors. Uh,
Saturation points, what exactly they are asking for the saturation points, because when we are using receptors for the talking purposes, in the simulation techniques, basically when the complex is we are using we are providing the temperature. Basically temperature is based on the alpha sheets and the beta alpha helix and the beta sheets. So it's depend upon the stability of your protein, whether the if the protein is having beta sheets more, then it will take stability will take the more temperature, and when it is having the alpha helix, basically, so it will take us less time for this particular simulation time. I think that this
I think
means
this is the standard.
saturation.
Yeah,
Yes,
preparation. No preparation wizards. We will get it. Yes.
yes. Next question. um, okay. What is the significance of taking three proteins and how they are comparable?
Yeah. So that's what. Again, it is based on our hypothesis, and definitely this VDR no, due to the presence of the co-activator, what I can say, its binding confirmations will be changed, and we wanted to test, though the VDR is vitamin D is present on the VDR, how this polyunsaturated fatty acids modulates. So that's what we have selected: three: wild type, mutated type, as well as with the co-activator peptides. So that is the reason we wanted to check.
The next question is on what basis we choose the candidate for the molecular dynamic, as here it is taken as
Yeah.
3VT-CDHA.
Yes. Yeah. You can. 1 minute. You can show the molecular docking results. So when we see the binding energies, no, this all the three compounds are showing a very good effect on all the three VDRs, especially this DHA. No, it is showing very good compared to the standard with normal, with coactivator, as well as this mutated. So on these basis, we have chosen DHA with 3BTC for our dynamic study. Yes. Energy, we have chosen the 3BTC DHA complex for the dynamic study.
Okay. Did you find any common structural changes between the bindings?
Uh, Common structural changes, that's what when we did the dynamics, no, we can observe, yeah, it initially, the active residues, no, do initial phase, maybe initial frames, no, there is a hydrogen bond interactions, the residues, the active residues, we had a little bit what I can drip. Then after that only they have attained the equilibrium. Definitely there will be a change in the all the three receptors.
Any other questions we have? Okay, what are the statistical symbols used in the molecular dynamics?
Statistical ensembles used in molecular dynamics. I think we have observed RMSD, RG, and statistical ensembles. Maybe I think Dr. Sakshi will take up this question.
Here actually ma'am has used 15 nanosecond ma'am. I think I am right for this one. 15 nanosecond simulation time by by fixing the temperature. As I told you that the temperature is basically based on whether your protein is having alpha helix and the beta helical beta sheets in that. And of course the RMST we are counting to check the, it's basically a root mean square deviation from each of the configuration. And then the radiation of gyration, as ma'am has already explained, it is basically utilized to check the compactness of your protein, basically the foldings and the unfolding mechanisms. So yes, here all these molecular dynamic which is this discovery studio is utilizing the charm dynamics, which is more accurate in the we having the different GPU modes and the CPU modes also. So GPU ones are more accurate than the CPU modes, basically faster in comparison with the time duration. So yes, they have done with the statical. These are the parameters which we have done in the molecular dynamics, which we have basically checking in these dynamic studies. So yes, I think we have done with our questions. So once again, ma'am, for joining us today for our, Women's Day webinar series. And it was very grateful to have you here with us. So thank you once again, and thank you all for that you could have joined us today. So thank you so much. Stay tuned with us for our next upcoming sessions too. Thank you all.
Yeah. Thank you all. Thank you once again. Yeah.